Among the general population, there is a lot of confusion about what exactly stem cells are. Due to the miscommunication between doctors and patients, many people believe that the only type of stem cells come from embryos. This is not the case. As you can see, there are many different categories of these cells that these cells can fall under. There are two main types of stem cells. Both are crucial at different stages of life. The first type is embryonic stem cells. The second is adult stem cells. Embryonic stem cells are the more commonly known type of stem cells. These are typically what come to mind when people think of stem cells. These cells are necessary in the development of embryos during the reproduction process because they are able to differentiate into a variety of cells. However, these cells can only be found inside a human embryo 4 to 5 days old as a blastocyst. There are many researchers and doctors in the regenerative medicine field looking at the possibilities these cells have in regenerative medicine. However, these are not the types of cells being used in the procedures done at regenerative medicine. The second type of cell are somatic adult stem cells. These cells can be found within the adult body and have a larger role as the repair and replacement team. These cells are undifferentiated, meaning that they have not decided what role they are going to have in the body. Some of these adult stem cells have a wider range of what they can do than others. Pluripotent stem cells, for example, can become many different types of cells. They are similar to embryonic stem cells in this sense. However, there are only a few throughout the body. Multipotent stem cells, on the other hand, can be found all over the human body and in abundance no matter what age you are. However, they are more restrictive in what kind and what they can do. Medicinal signaling cells, or MSCs, are considered to be a multipotent stem cell. Their importance to our procedure is that they reside in the bone marrow, and during the procedure, they are what recruit the signals, drugs, and proteins that are needed for the procedure to go well. The pharmacology produced by MSCs is what signals other cells in the body to repair and produce. The MSCs are collected when the bone marrow is harvested from the iliac crest of the hip. The bone marrow contains other valuable cells as well, but as the MSCs needed for the procedure. The bone marrow is then centrifuged, separating the bone marrow plasma nucleated cells, which are also known as the buffy coat, and the red blood cells. The plasma and buffy coat, which contain the signaling cells, are collected separately, and then the red blood cells are discarded. The plasma is collected and then passed through a special nanofilter that captures the anti-inflammatory molecules and eliminates the pro-inflammatory molecules. Finally, the signaling cells are mixed with fibrin to help with clotting. Part of the mix is injected beneath the bone where the arthritis started, and the rest is injected into the joint itself. Medicinal signaling cells have an amazing capability to repair and rebuild our bodies. It is exciting to see where research will take these MSCs in the future.